Horticulture and Landscape Architecture at Colorado State University. His area of research includes root exudation, root microbe interaction, including horticultural crops such as tomato, potato, etc., especially to understand phosphate and nitrogen cycling. As part of his research, he also interacts with producers and industries to develop products in the agricultural sector. I'm pleased to say that Jorge became an EMSL user starting in FY25, where he has an accepted large-scale proposal, where he's looking into the phosphate solubilization mechanisms between wild and domesticated crops. Please go ahead.
Vimal, thank you for this introduction, and I wanted to thank Vimal, Arunima, and also Amir for this invitation. It's a pleasure to be here. And I have Dr. Antisar and Mary Dixon joining me. They are going to be talking also about some of our research. So I want to give you a broad a perspective about our research, hoping that this will catalyze interactions in further interactions. So first of all, I want to put us in a context, and this is something that we started seeing around 10 to 12 years ago. Big agricultural companies such as Syngenta started putting a slogan in the internet and in conferences about giving importance to the soil. So these companies in the past used to sell seeds and fertilizers and pesticides. So this was sort of a new 10 to 12 years ago that companies like this started having conversations about soil. Furthermore, the CEO at that time of Syngenta started talking about giving importance to biodiversity in the soil. So all of this information started to appear 10 to 12 years ago, and catalyzed the purchases of a small biological companies. So there were a handful of companies around the world that were producing beneficial microbes. And these beneficial microbes were sold in agriculture, but in niche markets. So for example, AgraQuest was a company that they probably, their market was entirely California. And they sold at that time maybe no more than $10 to $20 million. This company was purchased by a Bayer for $500 million. Novozymes also got a big partnership with Monsanto. BASF bought another biological company that their market was entirely England. And as you can see here, they bought it for $1 billion. So all of these companies started buying small biological companies because nowadays we can see why. If you see at any big companies such as Bayer, BASF, Syngenta, microbes are part of their repertoire of products. Nowadays, farmers are encouraged not to be organic or to be intensive. They're actually encouraged to be sustainable. So they use fertilizers and chemicals and biological products in conjunction. So nowadays I don't think we can talk about being organic or being intensive. Now we are talking about in agriculture being sustainable. That is mixing all of these concepts. So let's talk about beneficial microbes. What do they do? So if you look at the catalog of a beneficial microbe, you can see that they have different activities. So they can increase nutrient uptake, so they can make roots more effective at taking nutrients. Beneficial microbes can also solubilize nutrients in the soil. It can actually fix nitrogen from the earth. So these microbes can help plants with also making nutrients available. As a result of this, beneficial microbes can increase plant growth. So usually when microbes are added to the soil, we can see anywhere from 10 to 20% increase in plant size, and in some cases the same in yield. Beneficial microbes can also suppress diseases. So some of these microbes are actually antagonistic against pathogens. So they could decrease using chemicals if microbes are used. What is interesting about these microbes is that in general, when you put a chemical treatment, you actually see an effect. So for example, there's a disease in a plant and you put a fungicide that actually kills the pathogen, but it kills it once. If we don't do anything, the pathogen comes back. Biological treatments such as microbes are very active, so they can have an effect, but this effect is longer, it lasts longer. So there's an interest in beneficial microbes because these are dynamic. So these are treatments that are self-sustaining. You can add it once, and you can have an effect for a long time. So these are the pros of using microbes. But there's also sometimes that these microbes, when we use it in the soil, they don't work, and that's why this technology has not been widely used. If you ask a farmer, they know that this exists, that this technology exists, but they're uncertain about whether it works or not. So sometimes it works and sometimes it doesn't. And sometimes it doesn't work because we lack knowledge about application timing, so especially in the soil, we lack knowledge about dosages. There's multiple phylotypes of these microbes, and sometimes these are specific to certain species of plants, so there's an interaction. pH and organic matter of the soil is important for establishing these microbes in the soil. And sometimes there's a poor screening conditions of microbes, right? So a microbe that was isolated in the tomatoes 20 years ago in California, for example, Bacillus subtilis, is now used widely in corn and in cotton and in different crops all over the world. So we are thinking that this species isolated in one place in California could be used everywhere. So in our lab, we think that there's something else in the soil that is happening, and that's why sometimes we don't see an effect. That is, that these beneficial microbes are actually competing with an endogenous rhizosphere microbiome that the plant already has. So I'm going to tell you a little bit about this last bullet. We've been working a lot and trying to understand the microbiome of soils and the rhizosphere of multiple crops in order to try to increase the use of biologicals in agriculture. So this is the area where we work. We try to understand the soil and the roots and the microbes. And but as all of you know, this is a very it's an underground environment. We don't really see what's happening there. We can predict based on symptomology of the flowers and the leaves, but we don't really have a grasp of what's going on underground because we cannot see it, right? And that's where your fabulous technologies will enlighten this. We think that the soil has three components, and we all know that the chemical that is actually nutrients, the physical that is the fixes of the soil, a structure mainly, but there's a strong component that is biological in the soil, and this for a number of years has been a black box, right? We know that is there, we know that exists, but we cannot manage it. We cannot understand it. So if we focus on this component, the biological, but now we add the root, so we have a root of a plant that is growing in the soil and is interacting with this biological black box. There's knowledge about root microbe interactions. So this is interaction between one root and one microbe. And for the most part, this microbe tends to be a pathogen. So plant pathology, it has 50, 60 years of information about how there's pathogens that affect roots, one-on-one interaction. But if we take a look at the soil now, with all of these sequencing technologies, we know that there's not one microbe, right? There's a microbiome growing there. So our root is on a daily or hourly basis interacting with a complex nature of microbes in the soil. Some of them are good, some of them are bad, and how the root is able to actually discern who's good and who's bad and what to culture or what not to culture. And that's what I like to show you in the next couple of slides. Another concept that you all know is the rhizosphere, right? The roots of the plants growing in soil are not interacting with the soil, are interacting with the rhizosphere, right? So in the rhizosphere are, as you know, 5 to 10 millimeters of soil surrounding the root. This rhizosphere is unique, right? The pH of those 5 to 10 millimeters, as you know, is unique. It's a different pH than the rest of the soil, different microbiology, etc. We know this as a concept, but I can tell you that this actually works in reality. So in South America, in Peru and in Chile, there's a large number of growers of grapes, table grapes. Most of your table grapes that you see here or you buy here in the winter probably come from there. And these grapes are grown in deserts. So you can imagine this as sand. That's when you go to the beach, and each one of these plants is actually being irrigated with a drip. And the drip is given the nutrients, the water, in some cases, the beneficial microbes. So if you look at these plants after 5, 10 years, these are table grapes, similar as what you have here around Walla Walla. They're in sand. But if you dig into the rhizosphere after 5 to 10 years, you actually find soil. So you see sand around, you dig sand, but as you get to the 5 to 10 millimeters, that's soil. The root is able to actually change that structure. So you can see it visually that the root is changing the rhizosphere. So the rhizosphere is incredibly important because that's the area that the root can actually sense. Right? That's where it's taking the nutrients, that's where it's interacting, that's where it's culturing the microbes. Sometimes we get farmers that ask, how do I modify my soil? Right? So they have a soil that has a pH 8 and they want to turn it into pH 7, and it's impossible, right? You cannot change the pH of the soil, but you can change the pH of the rhizosphere, right? That's what you can do. So if we go back to our roots in the microbiome, this is how we understand what's happening. You have an array of microbes in the soil, and the root is culturing some of these microbes into the rhizosphere. So to make it simple, some of these microbes, we call them neutral. We don't know if they're good or bad. We have microbes that are pathogenic or that are competitors for nutrients, and these are the red, and we have beneficial microbes. So in agriculture, if you look at the microbiome of plants, this microbiome tends to look like this. So more of the negative ones and less of the beneficial. And the challenge that we have is to turn it like that, to make it more beneficial and less pathogenic. So we've done multiple rhizosphere sequencing projects through the years to try to understand how the microbiome looks in different conditions. So this is a publication that we wrote about comparing the microbiome of intensive cultivated potato plants and compare it to potato plants growing in an organic way. And the microbiome is different, as you can predict. It takes usually 3 years to go from an intensive cultivation to a organic. If you look at the microbiome, so the microbiome takes time to actually adjust from an intensive to an organic. We have also done sequencing projects of plants in nature. So how the microbiome looks of a plant that is not cultivated, but is just grown naturally. And of course, we have done plenty of experiments in the greenhouse. So this is a study in which we have 2 model plants, Arabidopsis and Medicago, and we had soils of Arabidopsis growing naturally and soils of Medicago growing naturally. And we asked what happens if I grow the Medicago plant in a soil where Arabidopsis grew naturally, and what happens the opposite, So we have done a lot of those experiments, and based on that information, we have developed this very simple understanding, very simple, but I think is valuable. So if you look at the panel A, in the panel A, you see that there are 9 circles in the root. So these are 9 microbes growing in the rhizosphere of plant A. So this is a very simplistic understanding. So what you can see is that in nature, is the condition A is nature. You tend to have a diversity of microbes in the rhizosphere, but you also tend to have homogeneity. That means that you have 9 microbes, and all of those 9 are actually occupying one space. Okay. If you see in panel A, you see a microbe that is red. Okay, here's the pointer. You can see this red microbe, and for the purpose of this model, this is a pathogen. So you have a pathogen in that condition, but you have 8 other microbes that are actually keeping this pathogen under control. So that doesn't become pathogenic. It's there, it exists, but it's not doing any harm. Now, if you look at the condition C, in C we see a field or the microbiome of a field under agricultural conditions. What we usually see, we see that there's a few number of species, and also that there's a lot of heterogeneity. So that means that the few that are there are occupying most of the spaces. These spaces could be physical, or these spaces could be temporal. It could be timing. But that's what you see in agriculture, is a highly imbalanced microbiome. So now in agriculture, if you have a condition like C, and is creating a disease, you see how the plant is turning yellow. What growers do is they actually tend to put chemicals, right? In this case, let's assume that red is a fusarium, and the plant is a tomato. So what happens is that when that happens, the plant becomes yellow. There's a soil analysis in which we identify that that's a fusarium problem. We come with a fungicide that is applied into the soil. So what that fungicide does is takes the condition C to condition B. So the pathogen, as you can see in condition B, is highly diminished. These white spots is where the fusarium used to be. So the farmer controls it, brings it down, but there's always an inoculum. You can see here, this little red is there. The pathogen is there. So if the farmer doesn't do anything, this becomes a cycle. The condition B again goes to condition C, because the pathogen has the space to the little inoculum can take over. So that's why in agriculture we tend to have weekly applications of chemicals. In tomatoes in Florida, they actually tend to have daily applications of fungicides, basically to prevent the plant to go to condition C. You just keep it in B. So that's a cycle. So one of the studies that we found is like when is a good time to put biologicals, right? So when do you put biologicals in the soil? Beneficial microbes usually, for the most part, have to colonize roots. And the minimum condition that you have to give to the microbe when you apply it to the soil is to have roots. So one of the recommendations is put it when you have flashes of roots. Plants, especially perennial plants such as grapes, there's actually physiological flashes where you have lots of roots being produced. So growers put the microbes there. Another recommendation is before you put a biological, perhaps just put a light fungicide so you can lower your concentration of negative microbes, and then you put a beneficial. So there's a lot of recommendations now that farmers are using to apply the microbes. So these microbes, for the most part, are very expensive. So if you don't know when to use it, basically our farmers are just wasting money applying it. So soil health or root health is a word that we tend to listen often, you know. So for us, soil health from a microbiological perspective means this figure. Tries to have diversity of microbes, but also homogeneity. It's easier said than done, right? This is an easy graph to put, but this is where we are aiming, is that agriculture in the rhizosphere looks like this. So in order to get to this idyllic rhizosphere microbiome, we need to understand what's happening and how the root is actually communicating with the microbes. So imagine if you have a seedling. This seedling is planted in the soil, it's culturing a microbiome. As the seedling grows, the seedling has different needs, and the microbes change. You know, so as the plant is changing, the microbiome that the plant cultures changes as well. So how the plants do this? They do it by chemical signaling, right? So roots can secrete compounds, and these compounds can have different functions, you know, signals, substrates, and they can also be antibiotic, light antibiotic functions. So some years ago, we wanted to understand the interplay between plant development, root exudation, and microbiome recruitment. So we wanted to understand at the basic level how the plant changes the output of compounds as they grow, and how these output of compounds modify the rhizosphere microbiome. So we grew Arabidopsis in vitro in different stages of development, and we collected root exudates, and these root exudates were analyzed by chromatography, by GCMS. And I'm going to show you some information here. So this is a principal component of the secretion of compounds. In red, you have the seedling stage, and in green is the vegetative stage. So at seedling and vegetative, the plants are secreting similar types of compounds. Okay. When the plants are in the bolting and flowering, the compounds change, and bolting and flowering compounds tend to be similar. So when the plant is young, they secrete certain compounds. When the plant is getting older, other compounds are being secreted. What these compounds are, and this is what was quite interesting. When the plant is young, as you can see here, there's an abundance of sugars being secreted. You can see here. As the plant gets older, those sugars get minimized. They're secreted less. On the other hand, when the plant is young, there is you can see in red, amino acids are secreted in red, but as the plant gets older, those amino acids increase in concentration. And similar thing happens with phenolics. So we found that this was found by other people in rice, that when plants are young, they just secrete a lot of sugars into the rhizosphere. And as the plant gets older, those sugars decrease, but then the secondary metabolites or specific amino acids are being pumped. At the vegetative stage, as you can see here, there's a balance of compounds, you know, of everything is being secreted in similar quantities. So we wanted to correlate this information with the same developmental stages, but now the plant grown in soil. Okay, so similar developmental stages, plants no longer grown in vitro, but they're grown in soil. But instead of looking for the compounds, we wanted to look for the microbes. Let's see what microbes are in the rhizosphere at those developmental stages. Same thing, principal component, but instead of looking for compounds, now we're looking at microbes. And then we see that the seedling and the vegetative stage, the similarity of the microbiome. There's a similarity when the plants are young, have similar microbiomes. As the plants get older, the microbiome associated with the roots change. And there's a transition stage at the vegetative stage. Very simple way of looking at the microbes, you know, seedling and vegetative are the microbiome composition is similar. And then at the bolting, flowering, similar as well. If we take a look at certain microbes in that in those stages, you can see that when the plant is young, microbes associated with defense tend to be cultured by the roots. You know, it makes sense, right? The plant is young, they don't have ways of defending. They tend to accumulate microbes that can help with defense. But as the plant gets older, the microbiome composition changes. These are usually microbes related to nutrient acquisition. So this is, keep in mind that this is for Arabidopsis. It's a plant that can grow from seed to seed in 2 months, quick life development of this plant. So you can see now that some ideas are coming into place, right? The plants, when they're young, they tend to secrete sugars. And as we know, sugars are substrates that are used for almost every single microbe. So it looks like the plant at the beginning is trying to culture as much as they can, you know, bring into the rhizosphere as many players. As the plant gets older, the secondary metabolites are the ones that are maybe keeping the ones that are needed for a particular function. The trick here, if you go back to, you think about the model at the beginning when I showed you condition A and condition C, you know, so the plant, unfortunately, is not realizing that it's living in a soil that is not homogeneous, right? So if the plant, probably evolutionarily speaking, thinks that it's under condition A of that model, where everything is perfect. So it's trying to culture as many things as they can. Unfortunately, if the plant grows in that soil C, where the soil is unbalanced, all of those sugars is making the problem worse, right? It's trying to culture more of the bad guys. So that's something that we need to discern more. I think technologies such as the ones that you guys have will probably, will for sure help in this. So we, as a proof of concept, we did another study in which we wanted to understand what's the effect of root exudates in modifying the microbiome of soil. So we got rid of the plant, and we applied root exudates to soil without a plant, just to understand if we can see similar interactions as we saw with the plant. So if you remember, this is the output of root exudates at different stages of development. So we picked up the exudates from the vegetative stage because those had a balance of different compounds. And out of those exudates, we added those exudates to the soil for 4 weeks. And those exudates, we even dissolved it in different fractions. So the exudates, we extracted it, the water fraction, the chloroform fraction, the ethyl acetate fraction, and developed 4 different fractions that we added to the soil. And with that, we sequenced that microbiome with these exudates. And what it came interesting is that phenolics were actually driving this interaction. So phenolics were able to actually differentiate microbiomes more so than sugars, you know, so which we know these are signals, right? Those are the signals that plants use. So with this understanding, now we know that the plants can modify the microbiome based on the output of the root exudates. And we also know that the key players on this modification of the microbiome are phenolics, you know. So we are modeling or trying to understand basically this feedback between the root exudate and the microbe, especially with a microbial community, right? So some of the ideas that we are exploring is, for example, there could be a chemical A exudate that can decrease the concentration of microbial species. So this could be potentially a compound used to lower the amounts of pathogens, for instance. Or it could be a chemical B that could actually increase general diversity of the soil, you know? So it's a chemical that can be a signal and can have microbe interactions that can basically make the microbiome become more diverse. Or it could be a chemical C that can induce an interesting microbial species for a particular purpose. In this case, I highlighted nutrient uptake. So we have been digging deeper into this to try to understand if there are compounds present in the root exudates that can modify or induce phosphorus solubilization. And that's what I want to tell you next. I think we still have that. So why phosphorus? We started thinking about phosphorus around 7-8 years ago, because the use of phosphorus in the world, as you can see here in the graph to the left, has increased considerably through the years. So and this mineral comes, as you can see in the graph to the right, from 5-6 different mines all over the world. So these are companies that extract rock phosphate. What is becoming obvious is that as you can see here, that the demand for phosphorus is actually exceeding the production of phosphorus in the world. So this is a mineral that at some point will disappear, right? I mean, the mines, unless we find new mines, this phosphorus is probably going to keep going down. We work with two of those companies that produce phosphorus. One is called Haifa, that is based in Israel, and the other one is called OCP, that is actually based in Morocco. Those two companies are the main producers and sellers of phosphorus in the world, and they are also looking for ways that they can make the phosphorus more efficient, right? They know that the production will decrease, but they want to add something to their fertilizer to keep the same yield. So we've been having some of these ideas with them. And as you can see here, these are the main fertilizers that we use in agriculture. So phosphorus is an interesting, this problem is an interesting playing ground for all of the information that we have accumulated through the years. And there's something called the phosphorus legacy that is what we want to tap. So if you put fertilizer into the soil, like a regular fertilizer, this fertilizer, 10 to 40% is actually taken by the root. So the root takes it, and that's what that is the nutrient. Unfortunately, there is a 60 to 90% that gets actually trapped in the soil, and this phosphorus is what is called the phosphorus legacy. This phosphorus that gets actually trapped into the soil is unavailable to the plant. It's present there, but the plant doesn't have the means to actually use it because it's insoluble. So that's where our research is trying to understand, is basically what is the interplay between the rhizosphere microbiome, the root exudate metabolome, in finding ways to actually solubilize phosphorus. And that's the project that we are going to initiate here at EMSL. With that, I want to have Mary to talk about recent research on this space on tomatoes, and then Antisar will do the same on legumes.
Thank you. As Dr. Vivanco said, my name is Mary, and I'm going to be talking about some of the findings from current research coming out of our lab, specifically the findings related to these two publications listed here. My research follows the concepts about which Dr. Vivanco has been talking, those concepts about tapping into the legacy of phosphorus, how we can understand that by looking closely at the interactions and activity occurring at the root soil interface as driven by those chemical dialogues between the plant roots and the soil microbes. This research also has a big focus on domestication and wild species. We want to look at wild species because it allows us a fascinating opportunity to explore their broad genetic diversity. Wild crop relatives are extraordinarily genetically diverse, with the different colors in this illustration here representing that diversity. At domestication, however, we select for a subset of this diverse population, thus creating a bottlenecking effect wherein the genetic diversity sharply declines post-domestication. And now this bottlenecking effect has been observed in many crops, and in particular for tomato. In fact, current models estimate that tomato was domesticated from a population of only 300, which is far less than other crops. So when we wanted to look at how domestication and subsequent breeding events affected the ability of tomato to acquire phosphorus from that pool of legacy phosphorus and how it affected the ability of tomato to associate and communicate with the surrounding microbes. So to do that, we grew representative tomatoes from different domestication groups, and that includes 4 wild species, 4 traditional or heirloom tomatoes, which were developed around the year 1900, and then 4 modern tomatoes, and those were developed after 2020. They grew these tomatoes in phosphorus fertilized soil and in unfertilized soil, and the results are observationally clear. If you look at the wild tomato and the unfertilized group, the very top left 4 images, those are much smaller than the same species in the fertilized group. So just below that, you can see that size difference. Whereas the middle 4 and the right 4 tomatoes, the cultivated ones, they are much more similar. And you can see that statistically in our results on relative biomass. Now, relative biomass indicates the proportionate change in biomass between the unfertilized and fertilized biomass. So the higher the value, the less of degree of change. And if there's a lower value, that means that the plants are much bigger in the fertilized group. And here we can see that for both relative shoot and relative root biomass, the wild tomato was smaller than either domesticated groups, thus indicating that its growth was hindered by phosphorus deplete conditions. And this trend continues for shoot phosphorus uptake. Regardless of fertilization, the wild tomato accumulated less phosphorus in its shoot tissue compared to either cultivated groups. So its growth was hindered compared to the cultivated accessions. And when we look at the soil, we're also revealed that wild tomato had a unique microbiome. The microbiome, the rhizosphere microbial communities differed regardless of tomato domestication because of fertilization. And you can see that here with the shapes, which represent the fertilization group. So when you fertilize, it caused different microbial communities to form. But for the wild tomatoes, which is on the left side of this slide, you can see that yellow color representing Solanum pinelli, that cultivated a different microbial community. So in addition to fertilization causing a shift in bacterial community composition, that variety also caused a change as well. And those bacteria that were enriched in the wild tomato rhizosphere were beneficial. We saw a great enrichment, especially in Solanum pinelli, of phosphorus solubilizing bacteria. So even though the growth was hindered in wild tomato, it was highly associating with beneficial bacteria. And there were other interesting bacteria in the wild tomato rhizosphere. So this table shows PEG-identified predictive functions of bacteria that were significantly different between the tomato domestication groups. And what I'm trying to show here is that of all of the putatively beneficial functions, they're almost all more abundant in the wild tomato rhizosphere compared to modern and traditional. And then a co-occurrence network analysis also revealed an interesting characteristic of the wild tomato rhizosphere. In unfertilized soil, the network complexity and therefore possible interactions that are occurring at the rhizosphere microbial level are less for wild than traditional and modern, which had comparable levels of interactivity as shown by this network. But when we look at the amount of complexity between the two rows, unfertilized on the top and fertilized on the bottom, the wild network dramatically increased, and its level of complexity was now comparable to that of the modern traditional tomato. And the modern and traditional tomato remained the same between fertilized and unfertilized soil. So taken together, we found that wild tomato was different, it was markedly different from the cultivated accessions. Its growth was hurt, it suffered in its growth and development in conditions of phosphorus deficiency, but at the same time, its rhizosphere community was helpful, was beneficial, or at least associated with beneficial organisms. So what we're trying to do now in future and current experiments now is to utilize those interesting properties in the wild tomato germplasm and see if we can apply that to the varieties that are grown currently. And one of the ways that we can do that is by looking at candidate compounds and candidate bacterial species that can play a role in solubilizing and targeting those pools of legacy phosphorus. And those research questions are some of the focus of Dr. Antisar Afkairin, and I'll hand the mic over to her now.
So, hi everyone. My name is Antisar, and today I would like to present some of my projects. So, as Dr. Vivanco mentioned, and also Mary mentioned, we are trying to understand the mechanism. How can we bring the legacy of bee to be available to the plant? So this is the main focus for our research, and this paper has been published this year, looking for the enhancing of the phosphorus fertilizer in sensitive bacteria to enhance rhizosphere phosphorus bioavailability in legume. So we choose legume, as you know, all of legume is easy to grow, nitrogen fixer, and also have the availability to soluble phosphorus. So we choose different variety species from legume, like common beans, cowpeas, and peas, and we grow 14 variety. And we apply two treatment, which is triple superphosphate, which is the fertilizer, the phosphorus, and with control with nothing applying in. And it's worth to mention, the soil we are working in, it have 2.8 milligram per kilogram also be availability. So it's highly deficiency. And the nitrogen is about 3.2 part per million, which is the inorganic nitrogen, it's very poor. And we apply these two treatment to see how the plant will grow and how can we be able to identify some microbe or culture. The microbe have been solubilizing for B or mineralization for phosphorus to use them as a product or to increase or decrease the use of the fertilization. So as I mentioned, we grow different species, one of them the common beans, and you can see down there are different 6 variety from common beans. And also we grow cowpeas, and there are 3 variety for cowpeas. And we grow bees, and there are 5 variety of bees. And we put 2 treatment, which is fertilizer, TSP, the blue one, and the red one, which is non-fertilizer. As you can see with the common beans, Anasazi and cowboy, if you fertilize or you did not fertilize, the biomass is not significantly, but it seems like it's the same. So you're adding the fertilizer, you did not add the fertilizer, the biomass is going together, which is make it interesting. And cowboy also, and another one, is black bear. If you can see the black bear, the biomass is significantly different when you add fertilizer. So here it's giving me like the hints, when you are adding fertilizer for this variety, it grows better. If you do not add the phosphorus, it decreases. So variety matter between them. So we choose these 3 varieties from this species to go forward for do like some specific analysis of the microbiome. But coming up for the second species, which is the copies, and you can see the California number 5, it's the same. You add fertilizer, you do not biomass growing, and it look great. But CB4906, it's significantly different. So this species need more fertilizer. Similar with bees, Vale, the biomass is like similar. It's like it's not significantly different, but it's similar. But if you look for Marilosa and Cedrange, you can see they are significantly different, and these 2 varieties need fertilizer. So based on this, we choose these 3 varieties to go forward with the microbiome, and we did like the distant redundancy analysis for the microbial community composition here between 3 species, which is common beans, copies, and bees, based on the variety we choose for the biomass, and based on the fertilization and non-fertilizer. So as you can see here, between species, microbial community composition is significantly different. But between fertilizer, they are not significantly different. This is interesting things to see, honestly. After that, we went through different analysis. We did like the uptaking, and we did Olsen B, and we choose 3 varieties for this study, which is Anasazi from common beans, and have the correlation between biomass and the microbial community and the uptaken and the Olsen B in the soil. And all of them giving us this Anasazi is one of our elite. But when we check the relative abundance for this variety, we find all of these bacteria have been present for like mineralization and solubilization phosphorus. But these bacteria, when you add fertilizer, it gets depleted down. When you do not add fertilizer, it gets increased the abundance. So these bacteria, we call these bacteria sensitive for phosphorus. So when you add it, they will gone, or they will be reduced. But if you not, it will be highly abundant, and also they will increase the availability of phosphorus in the soil. And we saw this in the Olsen B when we did soil analysis for this variety. Second variety we choose it from, like the copy, is California number 5. This one interesting, honestly, because these bacteria have been presenting like the gene for mineralization and solubilization, but the relative abundance for both. If you add fertilizer, you don't add fertilizer, the bacteria grow and do the job. And it increases like the availability through Olsen B when we check it. So this is another variety, we call it insensitive for phosphorus, these bacteria. And Vale, another one, we choose it from bees, and they have also all of these bacteria have been expressing the gene for mineralization and solubilization. And these microbes, even though like they are not significantly different, so we call them insensitive for phosphorus. So they will not deplete as much as Anasazi with the relative abundance for these microbes. So the take-home message for this study is it's not only variety matter here, also the strategy for these plant and the microbe, how it can recover the phosphorus from the soil. And another thing is, as Dr. Vivanco mentioned in the beginning, he was talking about how can we reduce for using like the rock phosphate and save it. I believe these kind of microbes, they can be a good optimizing for the phosphorus and utilize it for future agricultural sustainability. So that's what I have today, and I am very thankful for all of you for listening for our presentation.
It was a great talk by all three of you. If you have any questions, you can. Okay.
I have a question, Mary and Antisar. For your guys' research, have you thought about identifying how the exudates change between the different plant types and, with and without the fertilizer and that kind of thing to see if that's having an effect?
No, we do
Yeah.
conduct re-exudate analyses in our labs too, and that is a future direction of our research.
Yeah.
So we just wrapped up growing wild and modern tomato in vitro and collecting re-exudates, and that's going to be carried on in this project that we're working on here. Yeah,
Okay.
so no, that's perfect question. Perfectly on our mind.
Yeah, and this is what we are thinking
Yeah.
about it. That's why we have like this opportunity with this funding to try to identify this kind of mechanism more deeper to understand the mechanism's way.
Gotcha. Thanks.
Do you all thought about the influence of the taking up more phosphorus and what that would mean to the ecosystem as a whole in a region? So for example, in Lake Malawi, there has been a lot of runoff in soils that have had a lot of fertilizer put on them. And then the lake system has really changed in its diversity, which has resulted in like less fish that the surrounding population can eat, which is their main source of protein. Yeah, I was just wondering if you thought like on that wider scale.
So is it like the eutotrophic mechanism? This is what will happen when like the phosphorus will run up and make this kind of issue for the environment.
Yeah.
We have not like touched this area, but it will be interesting to look at because this is one of the things.
Yeah.
It saved a lot of the runoff and also to reduce the impact of the ecosystem.
Yeah.
Yeah.
That legacy.
Exactly, yeah, because it's all come from the legacy. That's why the cycle of the phosphorus in the end, it will go where it will go running off and go to the ecosystem and
Yeah.
cause like the eutotrophics.
Yeah.
Yeah.
It would be interesting like,
Sure, yeah.
to see the calculation of like scalability of
Yeah,
what
sure, yeah.
happens in a small area
Yeah, for sure,
versus
yeah.
everything.
Yeah, it will be, yes, I agree.
I have a question.
I have a quick question. So when you do the comparative analysis, were those plants grown in different pots or they were grown in the same pots?
Well,
Then-
they're in different pots.
And across pots, we can assume that there can be microbial population differences, right? So were there any control experiments done to identify what were the populations in the soil to begin with, and then compared with the final results on how the presence and absence of plants changes and the presence and absence of phosphorus changes those microbial populations?
That's a good question. In the experiments that Antisar and I were talking about, we didn't sequence any baseline soil to see what the microbial populations are. What we were primarily looking at would be the differences in our treatment conditions. But in other experiments in our labs, it is standard practice to take a sample of bulk soil or pre-planting to find a baseline. But again, what we were really interested in would be how to examine just the change in these microbial systems as a result of different legume varieties for Antisar, different domestication stages from E, and then the fertilization. So more looking at the comparative elements in the rhizosphere of condition A and condition B, because we're more interested in as Dr. Vivanco was talking about, like the rhizosphere communities, and those are also starkly different from a pre-plant bulk soil condition.
Yeah.
Yeah.
Okay. And another thing that is just an observation and a follow-up question on that. I couldn't help but notice that the soil pictures that were shown in the presentation, some of those have very high algal growth, right? And from my experience, the presence of algal growth also affects the plant growth. And if I have to guess it, it might be because of the gas exchange or the airflow interruption, and that may also influence the microbial populations in the soil, right? And just an observation. I just wanted to know your views on that.
Yeah, by all means. Too much water, too much humidity in combination with certain plants can actually favor growth. I've seen the same brassicas tend to favor that kind of lichen or algal growth. And 100%, that's a member of the community of microbes. On the other hand, I mean, that community tends to grow in the very superficial part of the soil as compared to the roots that are deeper. But whether they can have an influence on their own cultivation of microbes or perhaps even compounds, I agree with you.
Okay. Okay. Cool. Thank you.
Mhm.
That was a great presentation that everyone, very informative. Thank you very much. So you mentioned, I think early in your slides that usually consider a rhizosphere region as millimeters around the root soil that are associated with root. But I'm sure you know that probably recently there has been some arguments about that rhizosphere can be even larger area, can be any area in the soil that kind of impacted by the rhizosphere, and it can be much larger than just few millimeters that are really attached or very close to the root. I just want to know what's your idea about that, if we can consider that, because it can be really larger effect in the
A good point. I think it depends on the soil too. So perhaps sandy soils, you might have a larger influence, more than the 10 or 20 millimeters. It can also has an effect on the watering of the system, right? So the compounds, especially water-soluble compounds, are released out of the root in those 5 to 10 millimeters. But if you have an area that is heavily watered, those compounds can move farther, right? If you take a look at the plants in cultivation, so in a field of corn, you will see that there's a rhizosphere for each plant, and there's plants that are 5 feet apart. So each crop has a rhizosphere. If you look in nature, if you go to a grassland, there's no separation between plants, and the roots of multiple plants are actually touching each other. So in that condition, you dig and you pull roots. I think that the bulk soil there is minimized, and when you're seeing a rhizosphere. So it depends on the condition, I think. The watering, the type of soil, the density of planting. So yeah, I mean, I think that 5 to 10 millimeter is highly academic. In nature, you may have more or less. Yeah.
Thank you.
Last quick question. Hi, I'm a fungal biologist, and so I wanted to follow up on some of the biocontrol treatments that you mentioned early in the talk. The question is, in addition to the treatment groups that you were looking at with phosphorus and fertilizer application, have you thought about seeing whether like the bidirectional nature of communication between fungi and plant roots might influence root exudate content in addition to other types of treatment?
If you want, I can answer that. Yeah. So we have not looked at fungi at all. And those are, as you know, a large member of the community of microbes in the soil. We haven't done that because the sequencing information is growing, but for the most part, you have a larger number of bacteria that you can get pathogen information. But for sure, I think the fungal community is a big part of the phosphorization ecosystem. We have a study that low mycorrhizae, but I'm sure that there's other fungi that are also playing a role there. We haven't looked at fungi at all, but I'm sure that that there are strong contributors to that civilization.
Thank you.